Hello and welcome to another video, Learning with Funke. Today we're going to be talking about glycolysis. And so for a quick background, what is glycolysis? Well, glycolysis consists of a series of steps, fundamental metabolic processes in which all living cells undergo. And pretty much you can think of it as we're starting with a compound glucose that has energy, and we are breaking down glucose to release energy as well as for creating metabolites that are useful in other metabolic processes in living cells. So glycolysis is coined from Greek words glykis, which means sweet or sugar, and lysis, which means splitting. And so what we're doing here is we're pretty much starting with a molecule of glucose, and in a series of enzyme catalyzed steps, we are breaking glucose down until we finally form pyruvate, which is a 3 carbon compound. And as you know, glucose is a 6 carbon compound. And so we start with 1 mole of glucose and we end in 2 moles of pyruvate. And so during these steps, sequential reactions of glycolysis, some of the free energy that was present in glucose is going to be conserved in the form of ATP and NADH. And just so you know what some important uses of glucose in the living cells can be, glucose can be stored in the form of glycogen, starch, can be, it goes through oxidation via glycolysis to produce pyruvate. Also useful in producing R5P and the pentose phosphate pathway. It can also be used, in producing as the starting monomer for cell wall polysaccharides. And so for a glycolysis overview, just so you know, glucose, for the purpose of this class, we're focusing on glucose to 2-pyruvate. For our next videos, we'll be going over, other processes. However, it is also important to know that under anaerobic conditions, you can go from 2-pyruvate to 2-ethanol in yeast fermentation. So this is fermentation to ethanol in yeast. And then you can go from 2-pyruvate to 2-lactate under anaerobic conditions, say like in your muscle cells where you produce lactic acid. However, under aerobic conditions, we go all the way through, glycolysis producing 2-pyruvate and go further down to the TCA cycle, and we end up producing 4 carbon dioxide molecules and 4 molecules of water. And we'll be going over the TCA cycle in a subsequent video. All right, so what are some key points of glycolysis? First, you have to know that glycolysis happens in the cytoplasm of the cell. And so for like the enzymes that we'll be going over and the reactions, all of those are happening in the cytoplasm of the cell. And so, like I said before, we're starting with a 6-carbon compound and we're ending with a 3-carbon compound called pyruvate. And this process consists of 10 enzymatic reactions. And in terms of the energy yield overall, it is important to know this. We have a net gain of 2 molecules of ATP. And ATP is it's a very key energy carrying molecule in cells. So it's important to know that we have 4 total, but 2 net ATP. It also generates 2 molecules of NADH, which is another energy carrying molecule, as we'll be seeing, downstream of the process as we go on in the lecture. And so glycolysis can also be anaerobic or aerobic. And so as we know, anaerobic means, does not require oxygen, while aerobic means, requires oxygen. And it can occur, so like I said, in the presence of oxygen, pyruvate can, undergo further metabolysis in the mitochondria, as we'll be going over in subsequent videos. That's not the focus for this video. However, in the absence of oxygen, it's also important to know that, like I showed in the previous slide, we can produce lactate or ethanol depending on the type of the organism. And so for regulation, yes, glycolysis is tightly regulated based on availability of glucose and energy in the cell. And so what we'll come to see is that several enzymes in this pathway, they have regulatory points. So like we have feedback control. I'll be giving an example towards the end of this lecture so you can further understand better. And so enzymes like hexokinase, phosphocryptokinase, and pyruvate kinase are all tightly regulated. And for the significance of this, it's very important. Glycolysis is very important because it helps us produce the necessary ATP, which are like building energy blocks that we need in other metabolic pathways. We also generate NADH. And glycolysis happens to be one of the, oldest metabolic pathways known. And so it seems to be like, it's pretty much traced to the earliest forms of life on Earth. So yeah, it's important to understand glycolysis and how the mechanisms involved. All right, so for aerobic versus anaerobic pathway, in the presence of oxygen, the pyruvate produced at the end of glycolysis is pretty much, goes through the citric acid cycle and oxidative phosphorylation, to further undergo TCA cycle reactions, which I'll be going over in the next slide. So please stay tuned for the next video that talks about the TCA cycle and how it works. However, the overall reaction is shown here on this slide. For the overall reaction for glycolysis, how we go from glucose to pyruvate, and then how pyruvate further undergoes oxidation to form acetyl-CoA. Acetyl-CoA then enters the TCA cycle, and here, NADH is used also in the electron transport chain, to further generate more ATP. Okay. And so for the anaerobic pathway, this, like I said before, happens in the absence of oxygen. Pyruvate from glycolysis undergoes fermentation, or it could also, undergo lactic acid fermentation. And these are the two, reaction pathways for that. And so we see that in these two types of fermentation, alcoholic or lactic acid fermentation, we are generating 2 NAD plus, NADH. In both types of fermentation, NADH is oxidized to NAD plus, and this is crucial for glycolysis to continue in the absence of oxygen. So what we'll see is that cells undergo, fermentation to regenerate this NAD plus, and in this process, NADH is oxidized back to NAD plus, which allows for glycolysis to continue producing ATP. This regeneration is very important because the supply of NAD plus, has to be maintained for glycolysis to continue or proceed. And so without this regeneration, glycolysis would halt due to, a lack of NAD plus to accept electrons, because this NAD plus molecules are electron acceptors. And so the NAD plus that is regenerated during fermentation is typically recycled back into glycolysis. And this recycling allows for, continuous generation of small amounts of ATP through glycolysis, even when oxygen is absent. And so in summary, for this slide, anaerobic respiration, NAD plus is used in the glycolytic pathway and is regenerated through fermentation. However, it's important to know that, this does not enter the TCA cycle, as the TCA cycle is not active under anaerobic conditions. Okay, so enough of the background and info. Let's go right into glycolysis reactions. So what are the glycolysis phases, the reactions, and the metabolites? So glycolysis, like I said before, it has 10 steps, two phases. The first phase is the preparatory phase, and in this phase, we're starting from glucose, and then we're phosphorylating glucose. I have images of the slides to come, but this is just a brief background. So we start from glucose, and glucose going to G6P, and pretty much overall conversion at the end of this phase, we end up getting G3P and DAP at the end of the preparatory phase. And then there's an intermediate reversible step between the preparatory phase and the next phase, which is the payoff phase, where G3P and DAP get converted to 2 molecules of G3P, and that's in step 5. You'll be seeing that subsequently. And the last phase, the second phase, is the payoff phase, and this pretty much starts from conversion of G3P, like I said in the last step, and ends in formation of pyruvate. So we end up getting 1 mole of pyruvate per mole of G3P. However, since we're starting with 2 moles of G3P, we'd end up with 2 moles of pyruvate. And this is coupled with formation of ATP and NADH. And so this is the preparatory step. I'll be going over each step subsequently for more details. But here we see that the first step is glucose going to G6P glucose 6-phosphate, and this is catalyzed by exokinase. This is the first primary reaction. So what do I mean by that? I mean, it's a phosphorylation reaction where glucose reacts with ATP, gets phosphorylated, and it forms G6P, with ADP being a product as well. When I say G6P, that's because it's phosphorylated at the sixth carbon. So we see here that we go from this carbon without phosphorylation. Now it has a phosphate group. And so in the next step, that is catalyzed by phosphohexose isomerase, and this is just an isomerization step. So you can see here, all we have here is just an isomerization reaction for G6P, starting from G6P and going to F6P. Next step, step 3, this is catalyzed by PFK1, phosphofructokinase 1. Here, F6P reacts with ATP to produce ADP. And then we get fructose 1,6-biphosphate. So what you see here is we started with fructose 6-phosphate, right? Only phosphate at the 6th carbon. But then it has reacted with an additional phosphate from ATP. And then the first carbon also gets phosphorylated. The fourth step is an aldolase step. We go from fructose 1,6-biphosphate, to G3P, and DAP, and that is catalyzed by aldolase. And then the step that leads into the payoff phase is pretty much catalyzed by triose phosphate isomerase, which is typically called TPI, and I'll be going over that shortly in the upcoming slides. Okay, so once we finish the marking the end of the preparatory phase, like I said before, this fifth step kind of leads us from the preparatory phase into the payoff phase. And so you see here that's catalyzed by this TPI enzyme, where we go from G3P and DAP, and then we pretty much make 2 G3P molecules. And the payoff phase of glycolysis pretty much yields the ATP and the NADH in glycolysis. So what we see is in the previous step, right, we used 2 moles of ATP, so here and here. However, let me take that back a little bit. However, in this step, what we see is that we pretty much produce what? 2 moles here plus 2 moles here, so we produce 4 moles. So we use 2 moles, but we produce 4 moles total. That's why we have a net of 2 ATP. So when you hear them say, well, from glycolysis we get 2 net ATP, that is why we get 2 net ATP. Okay. However, in the preparatory phase, we see that, we didn't have any NADHs or NAD pluses. Those pretty much come in the payoff phase. So here you see we have 2 NADH produced here. And so overall, we get, a total of 2 net ATP and 2 total NADH from glycolysis. Moving on. And so talking about the phase of pyruvate, I kind of went over this briefly. So pretty much we have 3 phases. And so the first phase, we typically talk about, pyruvate being oxidized and going through this aerobic conditions, going to form 2 acetyl-CoAs and going further down into the TCA cycle to form carbon dioxide and water. And then we also have the lactic acid fermentation here, which is pretty much, you can see that in the muscle cells where we form 2 lactate molecules. And then the third major route is pyruvate catabolism that leads to ethanol. And this can be seen in some plant tissues and microorganisms like baker's yeast, where pyruvate is converted under anaerobic conditions to ethanol and carbon dioxide. Okay, now let's go straight into the steps. This is a more zoomed in, view of what is actually happening. I've given an overview before, but we're looking now at the reactions and what's actually going on at each step. So in this first step of glycolysis, glucose is activated for subsequent reactions. So we see here that at carbon 6, at the position 6 here, that has an hydroxy group. This is catalyzed by exokinase, right? And we end up going from glucose to glucose 6-phosphate. with ATP as the phosphoryl donor. So ATP is the phosphate donor here. And this reaction is irreversible under intracellular conditions, catalyzed by the enzyme exokinase. And it's important to note that, kinases are enzymes that catalyze the transfer of terminal phosphoryl group from ATP to an acceptor. And so they are, a subclass of transferases. And so the acceptor in the case of exokinase is a hexose, usually, D-glucose like we see here. And this shows here, like, what typically happens. So we have ATP phosphate being donated to that 6 position carbon, and then we get phosphorylation on C6. And it typically doesn't occur here because we have a carbonyl group that cannot be phosphorylated. So yeah, here that's how we go from glucose to glucose 6 phosphate. Now, in the second step, still in the preparatory phase, we go from G6P that we produce in the last step to fructose 6-phosphate, and the reversible isomerization of G6P to F6P. So we are going from an aldose, which is G6P, and we are going to F6P. And yeah, so this is like a more in-depth view of like the steps that are involved. However, the overall step is just an isomerization reaction, where, we're going from, you see here, we still have this carbon and oxygen and phosphate group intact. However, we have like a rearrangement or an isomerization, the same number of carbons, the same, groups, just an arrangement where this carbonyl group moves. And here, this is the general idea. G6P, catalyzed by phosphohexose isomerase in the presence. And so for the next step, step 3 of the preparatory phase, now we're going from F6P that was produced in the last step, and we're pretty much going to fructose 1,6-bisphosphate, and this is catalyzed by PFK1. It is important to know the enzymes and what steps they catalyze, and so that is why I've shown all of this here. And what we see here is that, we had one phosphate group, right, here on carbon 6. However, PFK1 catalyzes that reaction with ATP in the presence of magnesium, and now we get an additional phosphate group on carbon number 1, right? So now we have phosphate going from phosphate at the 6th position to going to phosphate at the 1st and the 6th position. Sorry. Okay, and then that takes us to the 4th step of preparatory phase. Okay, and so in the 4th step of the preparatory phase, we go from fructose 1,6-bisphosphate, right? And now we're cleaving fructose 1,6-bisphosphate to form G3P and DAP. And so this is catalyzed by an aldolase enzyme. And here we form, as you can see, there's a cleavage reaction that goes on. The carbonyl group at carbon number 2 facilitates carbon-carbon bond cleavage to yield 2 3-carbon products via the aldolase catalyzation reaction. And so still at this step, now that we've formed G3P and DAP, just to understand the symmetry and how carbon arrangements pretty much go through from the fructose 1,6-bisphosphate to DAP and G3P. And so what you can see here is, based on the cleavage, right, your carbon 1, 2, and 3 can go here, catalyzed by this aldolase enzyme to form DAP. This is the first carbon here, right? If you're looking at the chemical structures, this carbonyl group is carbon 2, which is that, and then this 3rd carbon is just here. After it undergoes the cleavage, this just becomes CH2OH, right? Because it reacts with water. Okay, and subsequently you get your 4, 5, and 6. So you're literally splitting it in 2, right? The first part is DAP and the second part is G3P. Consequently, even though this is 4, 5, 6, you see here that this can also be, it can also be flipped about the y-axis. So this carbon down here could also be carbon 1, or, while this could be carbon, like you see here, carbon 2 and carbon 3, right? So, yeah. And so for the next step, the last step of the preparatory phase, here we have the interconversion of triose phosphates, so G3P and DAP. And so what happens is, because G3P pretty much goes into, the payoff phase, we have to convert that DAP to G3P. So we'd end up starting the payoff phase, which is the next phase, with 2 moles of G3P. And so what happens here is this enzyme triose phosphate isomerase, kind of like we talked about before, isomerase enzymes, they catalyze that isomerization reaction. It's typically like a rearrangement. And so what we see here is, so we go from DAP here, catalyzed by this isomerase enzyme, and then we form G3P. So you can see it's it's a rearrangement that happens in with the first and second carbon. And so we end up having an interconversion of the two products, for subsequent steps downstream. And that leads us to the next phase, the payoff phase. The payoff phase, the first step here is the oxidation of G3P. Now you have to remember, now we've converted that to 2 moles of G3P. And so everything I'll be talking about here is per mole of G3P. So because we have 2 moles of G3P, it's going to happen twice, right? So even though 1 mole of G3P converts to 1 mole of pyruvate from the slides that we're going over, you have to remember it's 2 moles that we're starting with. So we'll end up having 2 moles of pyruvate at the end. Anyway, back to the lecture. So we see here that G3P, reacts with an inorganic phosphate. You have to understand this isn't ATP, it's just reacting with an inorganic phosphate here. And the reaction mechanism is, always shown on the right. It's a more zoomed in version. So we start with an inorganic phosphate, and we also end up reducing NAD plus to NADH. And this is catalyzed by G3P dehydrogenase. And so we end up forming 1,3 biphosphate glycerate. So what does that mean? That means phosphate is reacting at we start with like carbon 3, but now phosphate reacts at carbon 1 to form, two phosphorylated, a compound with 2 phosphates, right? That's why we go from 1 phosphate here at carbon 1 initially, at carbon 3 initially in G3P, and then we end up having 2 phosphates here at carbon 1 and carbon 3 to give us this compound 1,3-BPG. Step 2, we have a phosphorus transfer from 1,3-BPG to ADP. And so that one of those phosphates gets transferred to ADP to form ATP, and now we form a new compound, 3-phosphoglycerate. So for the next step, here we're now converting 3-phosphoglycerate to 2-phosphoglycerate, and this is catalyzed by the phosphoglycerate mutase enzyme in the presence of magnesium. And so as you see here, the remaining phosphoryl group moves from, carbon 3 and then moves to carbon 2. And for the second to the last step of the payoff phase, now we have a dehydration reaction where we go from 2PG, 2-phosphoglycerate, to PEP. And so pretty much this dehydration is important to activate the phosphoryl for transfer to ADP in the last step, where we would eventually make pyruvate. And so here, you see here it's catalyzed by the enzyme enolase, right? We have a dehydration reaction, so we pretty much think of it as like taking out these two. So we have taken out hydrogen here and taking out hydroxyl group here to form that double ring, right? Which gives us phosphoenol pyruvate. And that will be, used in the next step to produce our final metabolite, which is pyruvate, as you would see. Okay, and so the transfer of phosphoryl group from PEP to ADP happens in this step. And so PEP, kind of transfers its phosphate group to ADP to form ATP. And we also form pyruvate in this step. And this pyruvate is what goes into the TCA cycle and further downstream to produce, even more ATP. I'll be going over that in subsequent videos. But it is important to know that this is also a very key enzyme catalyzed step, catalyzed by the enzyme pyruvate kinase. So pyruvate kinase catalyzes the formation of pyruvate. And this also happens in the presence of, metal ions, so like magnesium or potassium, and we end up forming pyruvate and ATP. And this marks the end of the payoff phase. And so while we're talking through all of these steps, it is also important to understand the importance of, it's important to understand that glycolysis is tightly regulated. And so we've talked about ATP being generated. We talked about the ATP used from glycolysis under anaerobic conditions being 2 ATP per molecule of glucose. Typically like way, way smaller, almost 15 times smaller than, under aerobic conditions when it goes through the TCA cycle and the ETC, electron transport chain, and oxidative phosphorylation, which I'll be going over in my next video, or videos rather. It's also important to understand that the flux of this glucose through these pathways that we've discussed has to be kind of maintained, right? In moments where you have too high energy, you don't want your body to keep producing ATP. And at the same time, in moments where your body needs ATP because you need energy to catalyze, like to drive some processes, you want your baby to be able to kickstart that glycolysis process to start making ATP. And so it's important to be able to regulate glycolysis tightly, and not just in terms of ATP alone, in terms of glycolytic intermediates, right? Because we talked about how pyruvate, acetyl CoA, these important glycolytic intermediates go into like other pathways, the ribose 5 phosphate pathway, the glycogen pathway, and all these other pathways that produce intermediates and compounds that are useful for our cells. And so, that kind of like also ties into why we need to tightly regulate this. And so, with all of this, you can see that if there's anything you've learned, you should be able to, you can see that there's some kind of complex interplay between, ATP being consumed and ADH being regenerated. And this all ties into, in terms of like the regulation, there's some, allosteric regulations of several glycolytic enzymes. An example of that, like I said before, key ones are like PFK1, pyruvate kinase, and you also have exokinase, which kind of like by modulating those enzymes, you can kind of affect the rate at which those steps are catalyzed, right? And that's the general idea here. And so, on a slightly longer scale, you can start looking at, glycolysis being regulated by even hormones from like, like glucagon, epinephrine, insulin, and by just by modulating like gene expression levels to control the availability and of these enzymes for catalyzing these reactions. And so glycolysis state regulation, here's a typical example. I use PFK1, of course there are more than one, but this is just a key example. For like allosteric regulation, some of the things that we can consider is like activation by AMP or ADP. So what we see is when the cell's energy levels are low, and that by that you would see maybe high concentrations of AMP or ADP or low concentrations of ATP. That kind of ties into allosteric activation of PFK1. And this is because AMP and ADP, like I said before, they can be indicators of low energy status, that signals the need to produce more ATP. And so by activating PFK1, you can speed up glycolysis to produce more ATP, right? Similarly, you can also be inhibiting PFK1 when you have too much ATP, right? So here, if you had too much ATP, it could go into the PFK1 step here, rather on step 3, and kind of like inhibit that when ATP levels are high, when there's too much energy, ATP can act as an allosteric inhibitor of PFK1. And so this can slow down glycolysis, because if this is inhibited, then we don't have this step going on. Otherwise, what will happen is we'll just keep going down the process and producing even more and more ATP, right? You don't want your cells to have too much ATP. because think homeostasis, you want everything to be balanced and appropriate. Okay. And so, other examples for allosteric regulation of PFK1, we also have an example of inhibition by citrate. Citrate is an intermediate from the downstream TCA cycle. This metabolite can also inhibit PFK1. And so, when we have high levels of citrate, like what does that mean? That means we are, if we have high levels of citrate, it means we have high energy levels because we are going all the way through TCA cycle, and that also means we're having too much precursors or biosynthetic precursors, and this can signal the cell to in an allosteric regulation to decrease glycolysis reaction, so the flux through glycolysis. Similarly, the other examples we have activation by F26 biphosphate. This is a potent activator of PFK1, and it's pretty much an important regulator in glycolysis. Its concentration is regulated by insulin, and that kind of like reflects the metabolic status of the cell. And so another example is fructose 1,6-bisphosphate, which we mentioned, which is here, right? The product of step 3. It can participate in a feed-forward stimulation mechanism in glycolysis. So if you're having fructose 1,6-bisphosphate, this typically has been found to activate pyruvate kinase. And if you remember pyruvate kinase, that's the enzyme that catalyzes the production of pyruvate, right? So it can activate pyruvate kinase in another enzyme in glycolysis for like the last step, like we talked about. And there are other mechanisms. We have covalent modification mechanisms that PFK1 can be involved in. We have gene expression mechanisms. So you would see typically in some scientific studies where we have transcriptional regulation. And so the expression levels of the PFK1 gene can be upregulated or downregulated. So, reducing the amount of like the activity of the enzyme to, accommodate and adjust to the cell's ATP needs or other metabolite needs, right? And so, and then another key part that I kind of spoke into is, integration with cellular metabolism. So, PFK1 sensitivity to ATP, ADP, and AMP can kind of help regulate glycolysis to ensure that we have sufficient energy requirements, not too much, not too little. And lastly, we have the feedback loops. So PFK1 is a part of a feedback loop. So that ties into, when ATP is abundant, it kind of slows PFK1 down. So here, when we have too much ATP, like I was talking about, it slows it down. So we're not going downstream to make even more ATP. And conversely, if ATP is, needed and ADP, we have, if we need ATP, it can also be activated, like I talked about before, kind of accelerating through glycolysis to make more ATP. And we've come to the end of the video, but overall, just to run through, the summarized output of glycolysis, we've said glycolysis is pretty much a key metabolic pathway present in living cells, right? And we start with a glucose molecule that pretty much is energy packed, and it ends up being oxidized in glycolysis to form two molecules of pyruvate with energy, conserved in the form of ATP as well as NADH, which undergoes further reduction downstream to produce even more ATP, as we'll be seeing in the downstream processes. When we start to talk about the TCA cycle, pyruvate oxidation, the ETC, and those further discussions. And next, glycolysis is tightly regulated, like we've talked about, with other energy yielding pathways to ensure that we're not having too much ATP or too little ATP. And all 10 glycolytic enzymes, like I said before, glycolysis occurs in the cytosol. So all these enzymes are in the cytosol, and there are 10 intermediates from the preparatory and payoff phases. Those phosphorylated compounds of 3 or 6 carbons, we see those in the cytosol. This is very important when we start to talk about the next phase, when we start to move into like the mitochondria and other parts of the cell where, we're seeing other reactions happen. It's important to remember that glycolysis happens in the cytosol. And lastly, for the net overall reaction for glycolysis, like I said, you start with glucose and you end with 2 molecules of pyruvate, 2 molecules of NADH, and 2 net ATP. You produce 4 total ATP, but because you invested 2 ATP molecules in your preparatory phase and you get 4 total ATP in your payoff phase, the net is 2, right? And that's why you see like the overall reaction here, which is also important to know, is that you get 2 NADHs and 2 net ATP. And so on that note, I hope, you've been able to learn, a lot about glycolysis, the glycolysis reactions, the key takeaways, the, tight regulation of the enzymes and, in terms of the energy levels and the metabolites that are produced. And I wish you good luck on your exams. If you have any other questions, please make sure you send me an email at learningwithfunke@gmail.com. If you have other topics you'd like me to go over, and I'll be happy to. Thank you very much and have a wonderful day.